Biological engineering isn't sci-fi. It's here, now. Scientists at Columbia University just demonstrated that it is possible to replace DNA segments in human embryos with hand-picked nucleotides, altering that embryo's genetic code at will. And it happened using CRISPR technology, a new miracle of science that has already created incredible cures, but also has the capacity to create the most destructive bioweapons ever conceived. And worse, this technology is something nearly anyone can use. This isn't a big red button in a military base. An environmental catastrophe could be unleashed by a simple mistake. But to understand why, we have to understand how it works. CRISPR itself isn't the technology, it's a feature of DNA. Clustered, regularly interspaced short palindromic repeats are specific patterns within DNA that guide RNA can essentially look up and find. The real breakthrough was combining this with an enzyme called Cas9. Cas9 is able to cut apart DNA, altering the genome of a cell. Scientists discovered that when you combine Cas9 with guide RNA, programmed to find a specific DNA segment, you can alter an organism's genome at will, with remarkable accuracy. Even further, after Cas9 chops apart the DNA, it can be allowed to heal itself, typically deactivating the gene. Or, the scientists can synthesize a new DNA sequence to be melded in its place, giving the organism a totally new gene. This is incredible technology. It allows doctors to create bespoke treatments for previously uncurable disease. Recently, a baby received unique gene therapy for a lethal metabolic disease. CRISPR saved his life. There is huge potential for creating not just one-of-one medicines for individual patients, but the ability to cure genetic diseases deemed uncurable. The FDA has already allowed gene treatments for sickle cell and beta thalassemia. There's no horror here. But this is just one part of CRISPR, called somatic editing. This is gene therapy for a living organism. A tissue sample is taken, the cells are edited in a lab, then put back inside to propagate. It only affects the cells in the targeted area of the body. But there's a second type of gene editing, one which permanently changes the genome of a whole organism. It's called germ-line editing, where scientists genetically modify an embryo. Because there are so few cells, the treatment can target all of them. That means that every cell in the growing fetus and eventually born organism will contain this genetic manipulation. And this isn't theory either. Someone has already edited human babies. In November 2018, Chinese researcher Jiankui announced that he had impregnated a woman with 2 gene-edited embryos. The purpose of the editing was to prevent the unborn twins from being infected with HIV from their parents. The embryos were edited outside the mother's body, then implanted using IVF. Fortunately, the twins Lulu and Nana were born healthy, but this didn't stop the immediate international backlash. He had performed the experiment without going through any ethics process. His peers called the experiment not only a failure of self-regulation, but misguided, premature, unnecessary, and largely useless. There are already well-known methods like sperm washing to prevent children contracting HIV from their parents, and they don't require nearly as much risk. For this mad science experiment, He was arrested and sentenced in China to 3 years in prison. But now he's free and continuing his research, with funding from a mysterious benefactor. As of 2022, the twins remain healthy, but independent researchers discovered likely errors in He's gene editing process, causing mutations. This means that for their whole life, the twins will undergo increased medical screening for the primary risk factor, cancer. But this gene editing wasn't somatic, it was germ-line, an embryo. And this means that even if these girls remain healthy the rest of their lives, the gene editing doesn't just have ramifications for them. When these girls grow up, if they have children, their CRISPR-edited genes go into them at the typical Mendel 50% rate. While this is cataclysmic for their family, it has small implications for the rest of the world. The genes will eventually be diluted out, especially if they have a negative effect. But imagine if this type of practice became widespread. Over dozens of generations, the human genome would become a hodgepodge of creation combined with human manipulation. In 1,000 years perhaps, most people could be in part molded by fallible human hands. Humanity could diverge from the intelligence of either natural selection or the divine. And we don't have to imagine this. They're already doing it to animals. Germ-line embryo editing is already being used all over agriculture. Fish species are being edited to grow bigger, faster. Cattle are being edited as embryos to never grow horns. Pigs are even being edited for xenotransplantation, organ transfers into people. But that's for another video. If you're like me, this is immediately horrifying. It calls to mind all these stories of playing God, like Frankenstein. Mutating animals for human desires is stomach-churning, all on its own. The opposite side would of course say, well, haven't we done this for centuries with selective breeding? Humans took the wolf to the dog, made the chicken triple in size, even altered all of our food over time to be bigger and more resilient. Someone with my Luddite outlook is labeled a bioconservative with all the pejorative connotation. And yes, it is true that we've been altering the plants and animals around us since humans discovered agriculture. But the difference here is that selective breeding is, well, selective. We choose from the natural outcomes of the breeding process to produce animals that have the best of their natural traits. Now with gene editing technology, we produce evolutionary level change all on our own. But some might think this is contained to agriculture. These animals are bred in captivity, kept alive by the artificial safety of ag tech. These gene-edited creatures will be constrained to human civilization since the gene edits would die out when in contact with nature and its brutal selection. Not quite. This is where I have to introduce another CRISPR technique called gene drive. Genes are in pairs, right? If we think back to 7th grade science, we can remember Gregor Mendel and his peas. He discovered that organisms carry pairs of traits, which means even if we see different amounts of expression, dominant, recessive, genes are still passed down in a 50-50. With CRISPR, we can change that. In germ-line editing, scientists can replace a gene to have 2 copies instead of 2 distinct options. This means the gene will always be inherited by the offspring. With this, scientists can forcibly alter the entire population of a species. With the assumption that an entire population interbreeds, in just a couple dozen generations, that modified gene would permanently alter the species forever. Of course, this is in a simplified scenario. Gene drives don't always pass down at 100% due to mutation errors. Also, population interbreeding can be constrained by geography, climate, or any other barrier. Further, if the new gene is too bizarre and causes infertility, early death, or in any other way prevents breeding, then it might die out before it manages to hit critical mass. But the technology does work. It's only been demonstrated in a lab, but that's only because it's deemed too dangerous to release a gene-driven organism, a GDO, in the wild. Presume that a GDO does propagate. There are plenty of plausibly legitimate beneficial uses for such an organism. A GDO could cull invasive populations of animals. It can make a species more resilient or vulnerable to pesticide. Or most promisingly, it could eliminate the ability for mosquitoes to carry malaria. We already have some methods of culling the malaria-carrying mosquito population, for example, airdropping infertile males. But a gene drive could permanently remove their ability to carry the parasite, without lowering their population. This seems like a good thing, right? You're saving human lives while altering the ecosystem less than nuking them with pesticide. It's a scalpel versus a shotgun. It would be an incredible public works project, preventing hundreds of millions of infections and hundreds of thousands of deaths every year. But even if that works as intended, there will necessarily be unpredictable consequences. Say the anti-malaria gene edit causes mosquitoes to die at a higher rate, of some consequence of the mutation. This means there's less food available for all the animals that eat mosquitoes, be they dragonflies, goldfish, mosquito fish, hummingbirds, bats, frogs, and all the others. That then causes a decrease in all of those animals' populations, which doesn't just cascade up the food chain, but into all the other roles they play in the ecosystem. For example, a decrease in the hummingbird population means less flowers get pollinated, which would then decrease the flower population. This then cascades out and out and out until the entire ecosystem is irrevocably altered, all because of one mosquito gene edit. Of course, this is built on an assumption I made, right? That something could go wrong. But the literature agrees. Gene drives present characteristics of all 3 types of emerging risks, like uncertainty, complexity, and unpredictable magnitude. Another paper simply says there is insufficient evidence to support the release of gene drive modified organisms into the environment. So that's it, huh? Too dangerous. We won't be deploying these GDOs because they could simply wreck the ecosystem with unintended consequences. And no one wants to destroy the ecosystem, right? Bioweapons have been used for hundreds of years. As early as the 14th century, Mongols were using plague corpses during siege. But bioweapons are typically limited in scope. If you deploy anthrax, smallpox, Agent Orange, it is approximately limited to where it is released. But things have changed now. With gene drives, you have weapons that don't just affect a whole ecosystem, but that cause permanent and irreversible damage. The same way you could engineer a mosquito to prevent malaria, you could potentially engineer it to be more susceptible. You could engineer a staple crop to be less fertile to produce an edible fruit. You could engineer an animal to be less productive and die out over time. All of these would produce the same cascading effect, but in the case of a malicious attack, you wouldn't be worried about the consequences. A GDO is no visually different from any other animal or plant. Since the editing is done on embryos, it'd be easy to dump tainted seeds in with an international shipment, or even get a GDO into a country with legitimate paperwork. It's also near impossible to track or blame such attacks. Citrus greening disease, or yellow dragon sickness, is a pathogen that destroys orange trees. It originated in China in the early 20th century, but reached Florida in 2005 and spread to a majority of citrus farms within 3 years. This disease devastated the citrus industry and massively increased the cost of orange juice. It's not known exactly how the disease reached the United States, but it theoretically came in on smuggled tree clippings. Now, this wasn't a biological attack, or at least it was never investigated as such, but the delayed reaction by nature makes it impossible to place blame. You don't know something's wrong until harvest. There's no way to see it coming. And a genetically edited organism has the potential to be even more destructive. On top of whatever initial harm it does, the effects cascade to the rest of the ecosystem. If Agent Orange is a bomb, a GDO is a nuke. There are already government programs dealing with this threat. DARPA's Safe Genes program is one of them. Scientists are working towards reversal drives, immunization drives, and other technologies that could theoretically mitigate a cascade. Of course, these aren't proven yet, and by the nature of GDO cascades, it'd be hard to deploy such a defense when you don't know it's happening until it's too late. But the actual counterpoint against why this would happen is the potential of splashback. The world is so immensely connected with globalist trade that the devastation of any major country's ecosystem would be a global issue. GDOs don't know borders, and ecological effects could spread unexpectedly to allies or even back to the attacking country. Because of this, my expectation is that this sort of biowarfare would be regulated like nuclear weapons. These GDOs are so destructive, they would have to be regulated under mutually assured destruction. But there's one critical difference. Nukes remain in the hands of state actors. They're the only ones with the resources to pull off nuclear enrichment and weapons development. Gene editing, on the other hand, is cheap, easy, and everywhere. Research papers already talk about the possibility of the unauthorized release of GDOs for not just malicious purposes, but for simple profit. A hapless actor might release a GDO for pest control, not knowing or not caring about the cascading environmental effect. Even more simply, a GDO insect could escape a university lab, propagating before they even know it's gone. Instead of mutually assured destruction between governments, it's mutually assured destruction between every mid-sized biotech company or research lab. Jiankui, with his mysterious benefactor, could do this all on his own. And no one needs to do it intentionally. They just have to make a mistake. And eventually, someone will. It's easy to imagine a mass extinction event, an ecosystem so out of whack that it ends most life on Earth. And obviously, whether it's nukes or GDOs, it's not out of the question. But the reality is, the current state of nature may be extremely delicate, but nature itself is extremely robust. An ecosystem is dynamic. It has inputs and outputs and balances itself based on the state it finds itself in. After a cataclysmic event, it integrates the consequences until it finds a new equilibrium. Even were things to go terribly wrong, life on Earth would not be destroyed, just changed. It's right to mourn the loss of species as we see them, even something as small as the golden toad native to just Costa Rica. Its disappearance is a stain against human intervention in the environment, a permanent shame we have to bear. But if we look outside of the human timescale, many millions of times greater than our lifetimes, we see the comings and goings of innumerable species, lost to time for better or for worse. If the dinosaurs had never died, we would have never seen the rise of mammals, the rise of humanity. Who's to say our absence wouldn't allow for something new to rise? So don't fret the destruction of the planet. At our worst, we can only destroy ourselves.
Oh my God, bro.